Hello, this is me, Eleanor, and today I'll be explaining the methods behind solving the structure of the cysteine-less intein. Now, your first question probably is, what is an intein? Well, the word intein is an abbreviation of interior protein. However, inteins are significant because they can splice together two separate proteins without leaving a backbone scar. Here, I am drawing a simplified split intein splicing reaction with the N-terminus intein in red, C in blue, and the two halves desired product, also known as exteins, in black and green. The split intein comes together and undergoes a mechanism that involves a cysteine and asparagine from the intein and another cysteine from the C-terminus extein. Shown here is the cysteine amino acid. Cysteines are actually a crucial part of stabilizing protein structure because two cysteines can react to form a disulfide bond. These bonds are stable unless put under reducing conditions, but this also means that cysteines are unreactive unless in such conditions. So if the intein requires a reducing environment to splice, the desired protein's disulfide bonds will break and the structure will fall apart. No structure means a loss of function. An alternative to cysteines are serines, which do not play an integral part in protein structure and are analog to cysteines. Recently, an intein was discovered that uses serines instead of cysteines. However, its structure and thus its mechanism is unknown. So my research focuses on the question, what is the structure of the cysteine-less intein? In order to determine its structure, I will be using a spectroscopy method called nuclear magnetic resonance, or NMR spectroscopy. To understand how this works, let's use an analogy. Humans are constantly having conversations with each other. We can only talk to each other at a reasonable volume when we are a few feet apart. However, we don't have to know each other or be bonded to hold a conversation. Atoms also have similar interactions, where they communicate with other atoms close by. Their conversations can actually be picked up by an NMR magnet. These magnets are massive, and one of ours is pictured here. Now, we need an unreactive and stable protein to determine the structure of. Ideally, the intein fused together with each of the three active residues converted to alanines. In order to isolate the protein, I add a solubility tag to keep it in solution and a purification tag to make it easier to purify. A cleavage site is incorporated between the intein and the tags so that the intein can be isolated later. In order to produce this protein in mass, a DNA plasmid that codes for the protein is put into E. coli cells. This plasmid is first assembled from pieces of other plasmids so that the gene for the inactive version of the intein is made. Upon induction, the cells go into overdrive, synthesizing proteins, including the intein. In order to separate the intein from whatever else is in the cell lysate, or the dead cell goop, is centrifuged and its supernatant is poured through a nickel column. There are nickel beads in the bed of the column that snag the intein but allow the rest of the sample to flow through. How is this possible? This is thanks to the purification tag, which has a strong affinity for the nickel beads. Once the unwanted junk has passed through, the protein is eluted by adding a competitor for the nickel beads, which push off the tag due to its high concentration. In order to ensure the protein is pure, the eluent is then run through an anion exchange column. The protein gets caught in the column and is reluctant to leave as a solvent is initially very low in salt. The concentration progressively increases for a couple hours until the protein elutes off at just the right salt concentration. The purity of the protein can be verified in a protein gel. Here, I've drawn an example of what a gel should look like with a sample from the unpurified cell, nickel column eluent, and anion exchange eluent. Now that the desired protein is purified, a number of NMR experiments can be run to identify the amino acid residues in space. For protein backbone assignment, about 3 different experiments are required to pick up different sides of conversations between the atoms on the backbone. These conversations show up in a spectra that is then interpreted and contributes towards solving the overall structure. Thank you for taking the time to watch my video. I am very excited to discover the structure of the cysteine-less intein, which will benefit protein chemists everywhere.